Welcome. My name is Stavros Ezekiel and I lead the microwave photonics research activity in the Department of Electrical and Computer Engineering and the Emphasis Research Centre. As the name implies, the Microwave Photonics Research Lab at the University of Cyprus is concerned with the interaction between the microwave and optical parts of the spectrum, and in turn the fusion of electronic and photonic components, mostly for applications in communications and radar. Now, by itself, microwave technology is known to form the backbone of wireless systems such as satellite and mobile communications, while photonics has enabled high-speed optical fiber telecoms that form the backbone of the Internet. In microwave photonics, our motivation is to exploit the massive bandwidth and the low losses of optical fiber in order to either improve the performance of microwave systems or to add new functionalities that would not be possible with an electronics-based approach. A good example of this is our contributions to 5G research through fiber radio, in which it is possible to transmit both the data and the microwave signals used for their eventual wireless transmission over several kilometers of optical fiber. Of added importance here is the fact that 5G and indeed beyond 5G networks will require large numbers of compact, low-power antennas and the ability to operate at much higher frequencies. Here, for example, we have collaborated with partners in the UK, Germany and Japan, setting a recent world record for the highest frequency of 94 GHz for an optoelectronic oscillator. Devices such as these are of interest in areas such as radar for the detection of foreign object debris on runways, or for linear cell-based high-speed train communications. Much of the work in microwave photonics has made use of the commercially available components you would find in high-speed fiber optic communications. This, however, is similar to what would have been the approach based on discrete transistors in the electronics industry of the 50s and 60s. What really enabled the electronics revolution was the development of integrated circuits, otherwise known as silicon chips. Similarly, in recent years we have seen the emergence of integrated photonics. Already the integration of components such as lasers and photodiodes on a single chip promises to revolutionize fields such as data centers and computer interconnects. In our lab, we have developed, through projects with the European Defence Agency and other European partners, a number of chip designs based on silicon nitride, which can be used to manipulate the spectrum of microwave modulated optical signals. Now, more recently, our focus is on applying the technology of photonic integration to space. Here, through partnerships with the European Space Agency, we are developing next-generation satellite payloads. This work is motivated by the small mass, volume and low power requirements that integrated microwave photonics can bring to this field when compared to the traditional technology of microwave waveguides, for example. I do hope that this short introduction has given you some idea of the work that our lab is performing in this exciting and interdisciplinary field of microwave photonics. Thank you very much for listening.
Molecules in our environment and in our body affect our health and our well-being. However, these molecules are too small to see. Our mission is to make sensors for molecular markers of disease in the quest to provide smart, preventative, and personalized medicine. My name is Chrysafis Andreou, and I am the principal investigator of the Nanotechnology, Imaging and Detection Laboratory. Our lab is part of the Department of Electrical and Computer Engineering at the University of Cyprus. Our main research directions are in the development of chemical assays for analysis of biosamples and in the molecular imaging of cancer. We develop technologies that help us form a bridge from the nanoscale, the size of molecules, to the scale of the human senses, in order to reveal molecules related to disease and then help in reaching a diagnosis and recommend an appropriate treatment. These technologies include microdevices and nanoparticles. Our nanoparticles are engineered to locate and identify specific molecules. In the body, the nanoparticles help us reveal the presence of cancer through medical imaging or deliver a pharmaceutical for targeted therapy. Outside the body, we use them for analysis of biofluids, for example, in the detection of viruses in saliva. Our micro devices can manipulate small amounts of liquids automatically and reproducibly, with very high precision, and perform chemical analysis quickly and with very little sample. We also use these devices for biological tests with cells or with model organisms like zebrafish. These type of devices are often called labs on a chip because they can perform many types of analysis on a small integrated unit. Additionally, we use computational techniques to simulate and optimize our systems and algorithms based on machine learning to interpret our results and make predictions for new samples. We have capabilities for chemical synthesis of different types of nanoparticles and for fabrication of microfluidic devices using 3D printed molds. We mainly focus on optical techniques like Raman spectroscopy and microscopy for imaging and analysis. Our main projects at the moment include the development of nanoparticles for imaging and treatment of aggressive breast cancer, diagnosis of urinary tract infections, and rapid testing for the coronavirus SARS-CoV-2 that causes the disease COVID-19. Our lab consists of 3 postdoctoral researchers, 2 graduate students, and a number of undergraduates. Most of our funding at the moment comes from internal university sources and the Cyprus Research and Innovation Foundation. Our main collaborators include the Biology Department and the Medical School at the University of Cyprus, the Cyprus Institute of Neurology and Genetics, the Cyprus University of Technology, and other labs within the Emphasis Research Center.
Monitoring of volatile organic compounds is of increasing importance in many application fields, such as environmental monitoring, indoor air quality, industrial safety, fire detection, and health applications. The challenges in all of these applications are the wide variety and low concentrations of target molecules, combined with the complex matrix containing many inorganic and organic interferents. Measurements of VOCs are becoming even more important due to the stringent environmental regulations and increasing health concerns. I'm Assistant Professor Arabios Agapiou, and I'm the Director of Volatolomics Research Laboratory. The VRL was established in 2016 at the University of Cyprus. Our lab is also a member of Emphasis, multidisciplinary center at UCY. My group is currently comprised by 6 members, postdoctorate, PhD, and master scientist students. VRL is a sniffing laboratory specialized to perform qualitative and quantitative analysis of odorous and non-odorous biogenic or man-made volatile components released in the atmosphere or in a closed environment. VRL core interests are the identification and mapping of volatile chemical signatures of human presence, emanating from expired air, urine, blood, sweat, and other biological excretions. The monitoring of indoor-outdoor air quality components related to human exposure, that is, environmental contaminants and stressors, the early diagnosis and monitoring of virus disease and metabolic disorders through the emitted signatures or their correlation with human metabolic pathways, the detection and removal of emerging pollutants from the environment, and the aroma profile analysis of food and beverages of Mediterranean origin, for example, carob, wine, olive oil, milk, nutrivolatolomics, flavoring, etc. VRL is mainly using hyphenated mass spectrometric instruments along with preconcentration techniques like solid phase microextraction and thermal desorption in order to achieve the very low concentrations of the metabolomic chemicals related to human exposure. For example, occupational exposure chemicals, environmental stressors, indoor, outdoor pollutants, dietary sources, smoking, vaping, etc. VRL is equipped mainly with thermal desorption GCMS and SPME GCMS analysis systems. Additionally, we have access to a broad analytical instrumental arena provided through the chemistry department, including LC electrospray MS, multi-TOF-TOF MS, NMR, etc. VRL collaborates with national and international academic research partners in Europe and the United States, focusing mainly on the analytical determination of human and food metabolites.
The quest for smaller, faster, and more efficient electronics and optoelectronics has generated a high demand for new materials with improved performance and functionality. My name is Grigoris Sitskos, and I'm the coordinator of the Condensed Matter Physics Laboratory at the Physics Department of the University of Cyprus. In my group, we used advanced optical and microscopy techniques to study novel materials. Central to our work is the implementation of steady-state and transient optical spectroscopy to investigate the photophysics of such materials and evaluate their potential for niche optoelectronic applications, mainly focusing on light emission and light harvesting devices. The optical studies are further supported by microscopy techniques that provide crucial information on the structure-property relationships of the studied materials. Much of the work is focused on solution process materials in the form of suspension in solvents. In the liquid phase, the materials can be easily processed into thin films using inexpensive coating or printing techniques such as those shown in this slide. The solution processability gives us also the opportunity to deposit such materials at virtually any substrate, including flexible surfaces, incorporates them and mix them with other matrix materials and composites. We're currently interested on the physics and applications of tiny semiconductor crystals known as quantum dots. Such nanomaterials have some unique and exotic photonic properties owing to quantum physics phenomena. Two projects that are coordinated by ECMP Lab are currently running related to quantum dot research. The first is a collaboration funded by a strategic €1 million grant of IDEC Cyprus, in which the University of Cyprus, the Cyprus University of Technology, ETH Zurich, and EMPA Swiss Federal Labs are participating. The objective of this large grant is to explore the physics and optoelectronic applications of a new, exciting family of nanomaterials known as perovskite quantum dots. The second project is conducted with an EU-funded Marie Curie Fellowship with a view of combining two very important technological families of materials, namely nitrides and semiconductor quantum dots, to produce high-performance emitters for displays and solid-state lighting. The ECMP lab is currently composed of 2 postdocs, 2 PhD students, 1 master's student, and 1 undergraduate diploma physics student.
Welcome to the Optical Diagnostics Lab of the University of Cyprus. When light interacts with human tissues or other living organisms, it is altered in ways that can reveal underlying pathologies or pathogenesis. The researchers, engineers, scientists, and students of the Optical Diagnostics Lab of the University of Cyprus are dedicated to harnessing the power of light for better diagnosis and more accurate prognosis of disease. This research aims at combining microstructural imaging with biochemical spectroscopic analysis and advanced machine learning and artificial intelligence algorithms to enable faster diagnosis and improve the survival of patients suffering from cancer or infectious disease. Optical coherence tomography, or OCT, works by detecting infrared light reflected from tissue and determines the depth of its origin within the tissue by comparing this light with the light reflected from a mirror. If the light beam is scanned over the tissue, it can be used to create three-dimensional images of the tissue microstructure. The Optical Diagnostics Lab has developed novel techniques to extract useful information from OCT images, such as the dispersion and index of refraction. These features, combined with advanced image processing, can effectively detect cancer in a variety of organs and possibly determine its aggressiveness in the patient's prognosis. In a pilot study conducted by the University of Cyprus and the Nicosia General Hospital, the proposed technique has been shown to detect cancer in the gastrointestinal tract with more than 90% accuracy. When light hits molecules in tissues, its spectrum of wavelengths or colors can change depending on the molecules it has encountered. The detection of light reflected at different wavelengths, called Raman spectroscopy, can be used to determine the biochemical composition of the material under investigation. Unfortunately, the light reflected back with a changed wavelength is very weak. However, the intensity can be enhanced if the molecules are in close proximity with gold or silver nanoparticles. The nanoparticles, which are more than 100 times smaller than a human hair, can be excited by a laser and vibrate, resulting in highly bright light as seen in surface-enhanced Raman spectroscopy, or SIRS. The Optical Diagnostics Lab has employed SIRS for the diagnosis of urinary tract infections. Usually it takes 24 hours, using urine cultures, to identify whether bacteria exist in urine and confirm the diagnosis of a urinary tract infection. Furthermore, it requires another 24 hours and another culture to identify the most appropriate antibiotic to fight the disease. The lab has shown that, using SIRS, the bacteria causing a urinary tract infection can both be identified and their sensitivity to antibiotics determined within 2 to 4 hours with an accuracy of more than 85%. In order to fully utilize the data generated by the lab, as well as achieve the most accurate diagnosis and prognosis, the researchers employ advanced machine learning and artificial intelligence algorithms to better explore, exploit, and understand the data. These techniques include support vector machines, ensembles, and deep neural networks. Please feel free to contact the head of the Optical Diagnostics Lab for more information or to learn more about exciting new research.
Hello, my name is Silvestro Vignodis, and I'm an assistant professor at the Department of Physics at the University of Cyprus. I will briefly present to you today our work on the spintronics and nanomagnetism laboratory. Future electronic technologies need to continue to provide ever higher performance devices while improving continuously their energy efficiency. They are also expected to underpin the highly anticipated quantum computation revolution during this decade and beyond. In order to meet these requirements, new physical principles have to be thoroughly studied and then employed in such technologies. Electronics, as we know it, has so far only utilized a property of the electron called the charge. Another property of the electron, the so-called spin, which is of quantum origin, has largely been neglected. In our laboratory, we study spin physics with the aim to identify methods of manipulating and transporting spin for device applications that will fuel future electronic technologies. This work is carried out on high purity nanostructures that we make using ultra-high vacuum techniques, and then we use a variety of experimental probes to study how spin behaves in such systems. Our experimental probes include optical or microwave magnetometry, imaging at the nanoscale, and spin manipulation using microwaves. Our current projects include transport and manipulation of spin in organic semiconductors for low-cost and flexible spintronic devices, and also controlling spin in nanostructures in such a way that we enable high-contrast imaging or probing of such structures at large distances. And this ability can be used to enable subsurface geophysical mapping for energy and environmental applications.